This is the second video podcast for Chapter 6 on Rangeland Hydrology. What I want to do in this video podcast is look at the plot scale water balance in the rangeland context and then scale up from there to look at a patch, focusing particularly on the riparian patch in an arid, semi-arid setting, and then look at the watershed scale water balance, again at a larger scale than what I already presented in the first podcast from Walnut Gulch. So in many ways, this is going to be similar to what I did for the tropics, but now we're doing it in a very different setting, and you'll see there's very unique aspects to how the tree scale water balance plays out. When we look at the rangelands, many of us are influenced by old Western movies that we've seen, cowboy kind of movies, where you have the sort of sagebrush, out there and rolling around in the wind. But in fact, that's not really an accurate reflection of semi-arid, arid conditions in the western United States. In fact, there were vast areas of grasslands that are relatively homogeneous in terms of the processes that they have and the plant associations that are there. Now, there always was some percentage of shrubs. And just as an illustration, on this slide, I'm showing three key shrubs that we'll be looking at a lot in this video podcast. Creosote bush in the upper right, tar bush below that, and then honey mesquite in the lower left. And you can see that these are generally conical shaped, especially the creosote bush, whereas when you get to the honey mesquite, it's more of a complete hemisphere in its shape. And so just as we've looked at the fractionation of water into through-fall, stem flow, and interception in other contexts, you can see that that would be influenced in this context by that canopy shape, together with the fact that there's no major trees, there's just these shrubs and then the ground. And in the case of the grasses, there's none of that. There's basically, a varying density perhaps of grass, but more of an underground matrix of roots as opposed to more of the individual root differentiation that you have here, shrub by shrub by shrub. So those are the things we need to keep in mind as we look at the water balance. Here is a map that illustrates what conditions were like historically. So in the upper left of the slide, it says 1858 is the year, and this historical map shows that more than half of this rangeland area, which is in the Jornado Experimental Range in New Mexico, more than half of it was grassland, which is indicated by green. You can see that there are sizable patches of mesquite and then tar bush with creosote mixed in, but there is a substantial contiguous patch of grassland. By comparison to that 1858 condition, if we look in 1998, it's totally changed. Now, there's very little grassland at all. It's almost all mesquite, or more than half anyway, for sure mesquite, with a large amount of tar bush and creosote. So a complete transformation from a grassland predominant system to a shrub-dominated system with only a very small amount of grasslands. The primary driver for this change is thought to be livestock grazing that took place in the intervening time. So then, as an ecologist or a hydrologist, one might wonder, is it possible to go back to the condition that was there before, or is this the way that it's going to be? Does going back mean just stop grazing and then nature will take its course and go back, or do you have to actively do something? And so this gets to the idea of the resilience. So has the system, it's a resilient system, and I know this isn't defined here on the screen, but I'm just giving you some ideas. A resilient system is one that can return to its initial state, like it hasn't changed to such a degree that it can't go back. But if the system has changed beyond what we might call a tipping point, then it's not going to be able to go back, and it might constantly change, or it might have reached a new state of equilibrium where the abundance of the populations and the species that are present are stable, but it's just a different state. Okay, so I want to show you a set of cases that are all related to this overall situation here. And the overall question is that you've got this very small amount of water available in this setting. So how does this shrub-dominated ecosystem function? Why don't grasses reestablish? Why can't they just come in and outcompete the shrubs? The approach to doing this study involved assessing a plot scale water balance, looking at the morphology of these different plants, both in terms of their canopy, which we can classify into conical and hemispherical, or their rooting structures, which I'll show you in a few minutes. Then assessing the scope and differences in root channelization of stem flow. So remember from the tropical lecture, there was the hypothesis that there could be so much volume of stem flow that could create erosion. In the semi-arid case, there isn't going to be so much stem flow to create surface erosion, but it could still be possible that that stem flow is channelized into the ground along the rooting system, because at the interface between the root and the soil, there could be pore spaces in there, macropores we might call them, that allow the water to flow a little bit more freely than just through the soil itself. And then finally, drawing on all those different concepts, we're going to look at how shrubs collect and retain nutrients. The tools that were used in this study included manual gauges like we've seen before for the water fluxes, looking at what are called the life strategies, so each individual species might grow, how each individual can grow for that species, plot scale water balances, nutrient patterns, like what are the concentrations of nutrients in terms of the spatial pattern of those relative to the positioning of shrubs. And then also they did a dye study. So dye is just what it sounds like, put some kind of colored liquid on the ground surface or in rainwater, sprinkle that down using what's called a rainfall simulator. So you're like, you just have these nozzles allowing the water to rain out onto the tree. And as that happens, then that dye, some of it will just fall in the soil, some of it might get funneled as stem flow, and then you just trench into the soil when the system dries out and you see where the dye is. So here's a concept. This isn't the data. These are two contrasting ideas of how this study might go. On the left is a conical shrub. So in a conical shrub, you can see that there is a 45-degree angle between the ground and this line. And the conical shrub is pretty much has its canopy above that 45-degree line. And so with that conical shape, water is preferentially captured in the canopy and funneled down as stem flow and then down into the root with root channelization to deep soil storage. So the zone A, that's shown by this arrow in the middle, is where rainfall is redirected to soil storage. B is the zone for which there's throughfall, which is unlikely to be free throughfall in a conical shape, but there could be some. Rainfall velocity is reduced because it's hitting the leaves or stem and then falling off. There is some water retention there. Now look at the hemispherical context. Here, there's a stronger differentiation between this A-B zone, and then at the fringes there's this dedicated B zone. So this is a place where water may fall out of the sky, hit a leaf or a branch, and then drip straight down. So a greatly reduced chance of stem flow because there just isn't enough slope along there, or it actually could be bent over enough that the direction of flow would be off the leaf, not towards the stem. And so the idea is that you would get a greater concentration of things that are carried in by that water or that are picked up by the water off the leaf on the ground. And so you see this thicker black block underneath those in that dedicated B zone of throughfall. When we look at the hemispherical versus conical canopies, there's also a concept of how wind would flow through those. In particular, a cone is still a very efficient shape for wind flow. So the wind is going to go around that, but also when it goes over the top, it's going to be able to dive back down and reconnect. And so you can see it's very aerodynamic. The wind can blow all around, and there's no eddies that have formed behind there, just a very streamlined shape. On the other hand, with the hemisphere, the top of that canopy is large enough that it can create a zone on the back side, what we call the lee side, under which there is an area where there's relatively little wind that hits that. And so the air over the top does not reconnect, and it creates a pocket in there, what we call an eddy. And so the idea is that whatever debris, litter, dust, stuff that's being carried with the wind, or that might be broken off or coming from shrubs, is going to have a chance to accumulate in the lee side of a hemispherical shrub, but that won't happen for a cone shrub. So that's the concept. Okay, so let's look at the typical plant scale water balance. We see the through-fall function similar to what we've seen before. The creosote bush has a wide range of through-fall that takes place, whereas mesquite has the highest overall percent of precipitation captured as through-fall. So nothing too surprising there. When we look at stem flow variability is again highest at lowest precipitation levels. So you see this kind of 1 to 3 millimeter range. You have very high range of stem flow, but that it tends to level out once you get above 5 millimeters of precipitation. The creosote bush has the most overall. You see the number here of about 10 with values up to 12 fairly consistently, whereas the mesquite is lower. There are a few in tar bush that get that high, but most don't. And then the study also found that the length of the stem and the angle of the stem controlled stem flow volume. So that angle factor is where you have to have a good slope to drive the water down the stem, so the force of gravity carries it. If the canopy gets too flat at the outer edges, then it can fall off, and that's what you see with the mesquite, because the mesquite is more hemispherical than the creosote bush and tar bush. When they trenched the different plants, they found very different phenomenon in where the dye was going. So here is a slice through the ground showing 2 examples from the creosote bush. So it's not so important how these are different from each other, more that these are 2 examples that we're going to contrast with the other species. So for the creosote bush, there are small roots, but there are many of them, and you can see water is funneled down. Some of it drops off, but that where you see these black splotches, that those are the areas of dye, and so the dye is the indicator of where water goes. So that's why I'm saying where water did this or water did that. So the dye accumulated right under the center of the canopy, and then it also made its way along the roots, and you can see that even more thoroughly here. So when you have a lot of roots, the water is distributing equally among all these different roots that are all about the same size. When we look at mesquite, there tends to be one larger root and then disproportionately small roots, and you don't see much dye along those really small roots. They tend to stay along the major root. And so there's not as much that is at the ground surface. It tends to be funneled down along that main root. And then finally, in the case of the tar bush, the root system is relatively small, so you see more lateral spreading of the dye away from the root rather than just moving along the root. So those are different patterns. The dye is indicating where the water is going to flow in those different systems. Now this is where it gets really interesting, is if we look at the chemical composition. So first we're going to look at the water and then the soil. So how much of different interesting nutrients are present in gross precipitation, through fall, and stem flow. So let's start with bulk precipitation. They just sampled the rainwater, and it had these amounts of these constituents: nitrate, total phosphorus, magnesium ion, and calcium ion. The units here are in parts per million, ppm, and if you're not familiar with ppm, then I encourage you to look it up. Just as you know a percent means 1 in 100, a ppm means 1 in 1 million. So they're relatively small amounts of these constituents. And so you can see the bulk precipitation has these numbers. It's not so important what they are. Here are the numbers for through fall and stem flow, and the question that we have as scientists is, are the numbers for through fall statistically different from the numbers for bulk precipitation? And then are the numbers for stem flow statistically different from through fall and from bulk precipitation? And that's where the letter comes in. Anytime you have a number that has a unique letter, it means it's unique. And so in this case, if we compare nitrate, stem flow is 0.87, through fall is 0.16, and bulk precipitation is 0.1. Each one has a different letter. That means that each one is unique. It's no surprise that 0.87 is different from 0.1. But it is interesting to see that they have enough samples to be sure, this is confidence at the 95% level, that 0.16 ppm of throughfall is different from bulk precipitation. So if the rain only has these amounts of these things here, and then the throughfall and stem flow have more, that means that that has to be coming from somewhere, because mass can't be created or destroyed, it has to be conserved. So that means that the water is hitting the leaf or the branch and it's picking it up. The fact that stem flow is substantially higher than throughfall means that the extra duration of the water being on the branching network down to the stem is giving it the time it needs to collect a substantial amount of those elements. Now let's look at the soils. Here the differentiation isn't as strong, but there are significantly higher concentrations. So if we look at the ambient soil away from the tree, we can see these levels, and you can see we either have no letter or C and B. When we compare that to the area under the canopy, you can see that there is differentiation for N, P, K. K is potassium, right? But there is no differentiation for carbon. There's no differentiation for magnesium and calcium ions. If we compare what's coming right under the shrub versus under the canopy, there's differentiation of the first three again. There's no differentiation of carbon, but there is a differentiation for these cations. So the specific conclusions of the study are that 16 to 25% of the intercepted gross precipitation is transferred to deep soil storage by stem flow and root channelization. And that's the really unique thing here, is that these shrubs have the ability to channelize the water coming down the stem, even if it isn't a lot of water, but to channelize it, pick up all those nutrients and inject it into the ground under that stem. Then that water is available to initiate growth in the spring, even in years where there's no effective spring or winter precipitation. So what they found that's really remarkable is that even after 5 consecutive years of drought, shrubs can persist fine, but grasses can't. And so there's this dramatic difference in how these different kinds of plants, the shrub morphology versus grass morphology, is able to manage or hoard their water. Shrubs concentrate it in a small area right around the roots, whereas because grasses are distributed and don't have those deep root networks, they can't do that. So stem flow water is nutrient rich and creates to what you could consider islands of fertility that develop under desert shrubs. And then away from shrubs, any wind that's blowing around is going to be able to erode the ground and create more destruction and keep things from establishing there, whereas the shrubs are these calmer areas that hoard nutrients and minerals both at the surface and then in the subsurface. It almost makes you wonder, how did grasses ever be there in the first place historically? I don't know what the answer to that question is. For whatever reason, the grasses were there, but and as long as they were there and dominant, they were stable. It may be that over time, shrubs would have come to win. Like as disturbances took place, natural disturbances, whether that's droughts or fires or wind throw, maybe there would have been more and more openings for shrubs to come in as it is. So it's not clear what is a restorative mechanism that allows grasses to get going. For example, well, maybe if you have an abundance of water, what if you have a decade or two where there's so much water that those capabilities of the shrubs no longer matter? Maybe that would allow those grasses to reestablish. And that brings us to this key idea. The idea that a set of populations of plant species, what we might call a plant association, changes through time. We use the word dynamic to mean changing through time. And we have these different population sizes, a relative abundance of these different species, originally say 60% grass and 40% shrub. That changes through time, and we can use the word trajectory to mean how the abundance of each population changes through time. The trajectory is the totality of those changes as it occurs through time. What we're seeing here is that how that occurs, like grass is going from 60% to 65% to 75% and shrubs going down, or maybe among shrubs, mesquite actually going up as others go down. That the set of those population numbers are the variables, and as all those variables change, that's a trajectory. How that trajectory works and goes in one direction through time is not often reversible along the same path. So the numbers, if they go up 1, 2, 3, 4, 5, they're not going to come back down 5, 4, 3, 2, 1. A system might be able to get back to its original state. So you start at state A and you change, now you're in state B. You might be able to get back to state A, but it's very unlikely that you're going to get back there the same way that you came. And indeed, many times there is no going back. And the reason for that is that climatic changes, land use changes, disturbance regimes, they can all happen in a way that's irreversible. So if those drivers or fundamental controls change, then that's the way it goes. So we use the term hysteresis to define a system that goes from state A to state B by one trajectory, and from state B back to state A by a different trajectory. So you end up in the same places, or very close to them, but you don't do it by going the same way. So many people have the misconception that if we now want to manage the environment to put it back into some historical state, that we simply have to reverse what happened. That's very unlikely to happen because the process and drivers of the change are no longer going to happen just in reverse. Maybe we can just erase the landscape and directly plant things to get the same way, but really at the scale of what people are trying to restore in the landscape, that isn't feasible. So we have to think about how do we nudge the trajectory of a system by nudging different driving variables or other human or natural influences, but we have to be mindful of this hysteresis phenomenon. Okay, let's start to scale up. So the next scale up, we're going to look at a riparian corridor, such as the one shown in this slide. Notice that we're seeing a river channel, and it's pretty darn dry, which is typical of an arid context. And we're interested in seeing, over the course of a day, what is the spatiotemporal pattern of water vapor above the riparian zone along a river. Here we're going to return to Walnut Gulch, and I didn't have this map before, but this shows a map of Arizona. Here's the southeastern region where Walnut Gulch is in the San Pedro River. And so here's the San Pedro River with some other things positioned on the map. And then here's the Walnut Gulch watershed again. Okay, so the technology that was used in this study is called light detection and ranging, or LiDAR. This is where you shoot laser into the sky, start from the ground, like mounted on a truck. You shoot it up into the sky, and as that happens, the energy gets scattered, and some of that returns, and then based on the nature of what returns, you can determine the properties of the air. Some of you may be familiar with the idea of using LiDAR to measure the topography of the landscape. That's a different use. In this case, we have a ground-based LiDAR shooting up into the sky and using its return signal to detect aspects of the air. So in this case, I'm showing you a map. The map shows a LiDAR in the upper right as stationed in this position. And then the colors on the map show different species. These black lines running from top to bottom show the boundary of a riparian zone. Inside the riparian zone, we have predominantly cottonwoods, but also some grasses and a little bit of mesquite. Outside of the riparian zone, we're dominated by sacaton and mesquite, and there's also an agricultural fallow area at the bottom here. So the LiDAR shoots a beam across, and then it can rotate, shoot another beam, rotate, shoot another beam, and so on. And so we're going to just look at one of these beams. There are a lot of other things that were measured in the study. Eddy covariance is another way of getting at evapotranspiration, wind observations. SODAR is a technology for measuring wind profiles up through the canopy. And then sap flow, of course, we've already talked about. So I'm going to show you a series of maps that are going to step through the daytime for a few hours. So starting at 9:13 a.m. on August 11th, 1997. And along the x-axis, distance from LiDAR. So the LiDAR is far off to the left, and we're already 250 meters away. At 350 meters away from the LiDAR is the fringe of the riparian canopy. Notice this dashed line. This shows the canopy in terms of low-lying shrubs and then the tree canopy of the cottonwoods. And that canopy comes out compared to where the stem is. And then it goes across this line. So this line is the top of the cottonwood canopy. The y-axis in this is altitude, so it's just height above the ground in meters. The colors that are shown on this map show the amount of moisture in the air. The black at the top is an area that's not detected by the LiDAR, so it's irrelevant. Bright red indicates the highest moisture, and then dark blue or to purple indicates a low moisture. So if you're red, green colorblind, this, I have to apologize, but it's not going to be clear what this map shows. But in general, you can see that in the cottonwood corridor, we're fully saturated with moisture. And then as you get outside of that, there's decreasing moisture away, but it's not a simple, like diffusion function. There are these blobs of moisture that are like going up into the air. And this is similar to a model that I showed in some optional slides on evaporation several lectures ago. So now let's go to move forward an hour and 15 minutes. And I'll just cycle back, forward, back, forward. What you can see as we transition is a dramatic reduction in moisture just in this hour and 15 minutes in the morning. First, the size of the cottonwood bubble of moisture shrinks, but within the cottonwood corridor, you don't see any significant change. It's still fully saturated. But outside of that, a dramatic reduction to where there's almost no moisture in some areas, and the blobs have substantially gone down. By 12:40 p.m., that's a very, very abrupt diffusion-like threshold just outside the cottonwood corridor, and the vast majority of the canopy is just bone dry. And then that continues. There are some individual blobs that you can see that come off as I cycle back and forth through time. So just in summary, you start in the morning, there is moisture being supplied by the cottonwood corridor to the surrounding region, but as time goes on through the day, that disappears. The riparian corridor itself remains full of moisture, but away from that, it's not. This next slide here shows a horizontal slice. Like if you were to cut off the top of the canopy and then look down into it, then you would see this. And in this case, I'm not going to show through time, but just the time average abundance of evapotranspiration. And there are contour lines here, so if you pay attention to these, you can see these are in units of 0.3 millimeters of water. And here is the scale for the color scheme. So the reds here are more than like 2.7 millimeters of evapotranspiration, probably per day. Well, no, that's way too low. That's probably per hour. And then you can see that there is a higher abundance overall in this canopy, and then it diminishes away from that. But you can see that even within the canopy, the rate of evapotranspiration does change. Okay, so we're going to scale up again and look at an entire semi-arid basin. And now we want to put this riparian zone situation into context. So we can see that there's a huge amount of moisture in the riparian corridor, but the river itself is bone dry. So that means that the moisture has to be coming from evapotranspiration, and the extent to which that moisture can really stay there versus like blobbing off and heading up into the atmosphere and being lost, so to speak, is really what we want to know. So if you have a river like the Rio Grande, which is shown in this photo, and that how much of the water in the Rio Grande is being taken up by transpiration versus open water evaporation or soil evaporation, because what you really need to know is as the water moves from point A to point B, is there going to be as much water at point B in order to meet human needs and environmental flow needs. Of course, we know that there are transmission losses. We've already talked about that. We would like to now understand where those losses are going. Before I focused on the groundwater aspect of those transmission losses, but that's really an incomplete story. So here we're going to do a river scale water balance to try to get at that. This shows the section that we're going to be looking at. Ottawa Bridge is the inflow, and then Elephant Butte is the outflow. The shading here indicates relative elevation, with black being higher elevation and white being lower elevation. And then these are just different points along the way where different things happen. We're looking at about a 40,000 square kilometer area here in the middle Rio Grande. So specific objectives were to describe the major components of a water budget for a semi-arid basin and river corridor, provide estimates of growing season ET, examine shallow groundwater responses to ET, look at the land use changes, and I'm not going to be addressing that, and then consider ways to scale ET measurements when you do it in an individual plot to look at how that would affect a whole corridor with this scale. The approaches used here were to take advantage of what we have as the U.S. Geological Survey, or USGS. They are responsible for measuring flows in many important rivers around the United States. Now, over time, our country has been defunding this network of stream gauges, so I think we've lost about 30% of all the stream gauges in the U.S., but still, they play a very important role both in science and in society. And so in this case, there are USGS gauging stations at Ottaway Bridge as well as at Elephant Butte. And so you know how much is coming in and how much is going out. So you know how much you have to account for is the difference between the two. To measure evapotranspiration, they use what are called flux towers. That's similar, either using the Bowen ratio or the flux tower. You're actually measuring the wind as it's carrying off that moisture, and so you have an energy balance. You've got the direct measurement of flux, and so that's all taking place. Field mapping of leaf area index to try to see if there's a relationship between LAI and ET. And then also characterizing land use by satellite imagery, so that if you knew something about leaf area index and land use, you could then infer ET. Here are the inflows and outflows. The x-axis is time from like 1990 to 2001. Y-axis, mean monthly discharge in cubic meters per second. The upside down closed triangles are the inflows. You can see they spike higher, and the base flows are higher, so that overall the inflows at Ottaway Bridge are high, and then the dashed lines with the open triangles are Elephant Butte Dam, basically where the water is accumulating. And you can see that the peaks are diminished, but more importantly, the base flows are dramatically changed. Here's the evapotranspiration measured at one site in 1 year. Of course, you can see this cycle from day to day. There's variability, but overall there is the seasonal cycle like we've seen before. This shows the relationship between evapotranspiration and leaf area index. Now note, there are not enough points here to draw a regression line. So it isn't a relation in the idea that there's a function that we can fit here. Please never take 4 points and try to fit a regression line to that. We could simply say that for a leaf area index of 3.5, that we have an evaporation rate of about 7 to 8 millimeters per day. And for a leaf area index of 2.5, we have anywhere from 5 to 7 millimeters per day. So there is a clear differentiation between these two leaf area index, but we really can't create a function from that. Okay, so this shows a classification of the river and riparian corridor species. On the upper left, colors correspond to different species plant associations. In the lower right, we have the leaf area index that's presented. And then finally, here's the water balance. So we start with 1,419 million cubic meters of water per year flowing past Ottaway gauge. And then at the other end, there's no specific number, but a range of 370 to 1770, whereas flowing in, we had a range of 370 to 2714. But looking at the average number, we start with 1419. There are some tributaries and municipal inflows that come in, so you can sum all those up. The river itself experiences open water evaporation. Irrigated agriculture is pulling water out, pumping it out for use in agriculture. The shallow aquifer is discharging water to the river, but it's also receiving recharged water to the aquifer, and in the net it's receiving more, so that's the transmission loss. Of that shallow aquifer, riparian species are then sucking up a substantial amount of water, more than the net exchange that's there for riparian ET. And then downstream of the Santa Acacia gauge, we see a substantial amount of a combination of riparian ET, agriculture, and open water evaporation. And then the reservoir at the end here also has as much evaporation as any of those. So in summary, evapotranspiration accounts for 20 to 50% of total depletions from the river. Dense stands of salt cedar and mixed cottonwood, salt cedar and Russian olives had the highest rates of annual evapotranspiration, which could range from 100 to 120 centimeters of water per year. Our first order river corridor estimate of total riparian zone ET was 150 to 250 million cubic meters of water per year. That's a huge amount of water that's, I don't want to say being lost, but it's certainly being used. It's an ecosystem function that's making use of that water. The consensus opinion of the scientific team who participated in the balance is that open water evaporation, riparian zone ET, and irrigated agriculture represent the three largest depletions along the river corridor. Okay, so that gives you three different scales of thinking about what the water balance is like in rangeland, semi-arid, arid settings, and it also gives you a sense of the interactions between biota and water and even nutrients in those settings.